Excuse me. Yeah. So I guess two little things here. So most ovarian cancers, there is usually a high correspondence to BRCA1, not BRCA2, BRCA1. However, at least when this article was posted, there's no family clustering for ovarian cancer. So as opposed to where we see a very high, where it runs in families, there is a smaller sort of significance of that we see in those ovarian cancers compared to breast cancer. Or sorry, ovarian cancers with patients that already have breast cancer. Apoptosis, programmed cell death. This is the natural thing that cells want to do. If DNA is screwed up and don't have my proteins the right way, stop it, then let the cell die. One of the hallmarks is we lose apoptotic signaling. So apoptosis. You guys have to cover this for biochem? Is that this exam? Is that like the first or second? Oh, I think it's on this exam. You guys are going to get it maybe tomorrow. It's exciting. So apoptosis. Basically here, you have different ways this can occur. One of the, remember, if you see a P in a number, assume it's for halting cell cycle and or apoptosis. So P53 is a transcriptional regulator. Think of it like a TF. It's going to be changing other gene levels of other genes. Basically, what P53 does, if I have P53, it's going to upregulate P21. Another, once again, P in a number. And then that one then is going to be a CDK inhibitor. So CDK is those proteins that are part of all those other signaling cascades. So if I inhibit any of those CDKs, I'm typically going to be inhibiting cell cycle progression. I'm going to halt it there. Those P53, P21, they're going to stay high if DNA is screwed up, the protein is screwed up, and then once it goes back to normal, those levels go down, and then I can then go back into those signaling cascades. But if I don't repair those stay high. The end result is they're going to lead eventually to apoptosis. So, and also a lot of these P50, these P1s, they're also going to help with DNA repair. So they're going to be high, and because they're going to see, oh, DNA is screwed up, they're only going to pause DNA, but other signals along that pathway are going to allow for upregulation of maybe MMR or RR or other repair mechanisms to try to repair that DNA. So we do that, but if we have inhibition and we delay these pathways for too long, we delay S phase, but eventually then they're going to lead to signals responsible for apoptosis. And once again, I think everyone knows why we want to prevent replication, because then we have the bad DNA, we don't want it to go somewhere else. So you've got two different pathways for apoptosis. You have the intrinsic and the extrinsic. Extrinsic just means the entire thing's occurring outside, as in, example here is fast ligand. Remember, that's where the T cell sees your screwed up cell, and it's going to bind to it and just basically blow it up. It's going to bind to it and then cause apoptosis in that cell. Why do we prefer apoptosis versus necrosis? It's controlled death. Necrosis is shooting a nuke off in the cell, and you've got shit everywhere in your extracellular matrix, and that's not good. All right? Apoptosis, it's more refined. They kind of go in little bubbles, and then they just kind of kill each other off little by little. It kills itself off little by little, but you're not going to have shit all flying with other cells. Have debris in there. So in the case of the extrinsic, both of these are apoptosis. The extrinsic pathway is just another T cell helps out the other cell. It should help out the other cell and allow that cell to kill itself. Intrinsic pathway is it's going all on inside the cell. There is no ligand binding to the receptor. In this case, you're going to have high levels of P53, which we already had. Number one, going to delay, don't go to the next phase. But if we can't get our DNA to be nice and no mutated, non-mutations, then this is going to increase BAX genes. BAX genes, those, in general, for their main function here, high BAX genes are going to lead to apoptosis. It's going to lead to cell death. So high BAX genes are going to lead to a high level of caspases. Caspases then literally eat away at parts of the cell, and they're going to destroy the cell. I think on the next slide I have a little more. Yeah. So P53, once again, what did we say? It's a transcription factor. High P53 not only makes the high P21 to help with delay, but P53 itself will bind to a promoter region on the DNA and allow for transcription of the BAX genes. So you get these BAX genes. The BAX genes is going to then produce whatever the protein is, the BAX protein, and eventually where this is highly expressed is on the mitochondria. So the mitochondria are going to end up basically getting a lot of this protein, and it's going to destroy the mitochondrial membrane. And a lot of those caspases and other things are going to be located inside the mitochondria, and they can then leak out into the cell. So the mitochondria kind of gets blown up a little bit, and then all those all these proteins are going to degrade the cell membrane and the parts of the cell. They then leak out of the mitochondria, and then they destroy the cell. This is all part of the intrinsic pathway. Both intrinsic and extrinsic have caspases, but there's different numbers for intrinsic and extrinsic. Don't need to know that for blank, but unless they change biochem, you have to know those for the biochem lecture, either tomorrow or on Wednesday. Cytochrome C is one of the important ones as well, that'll get released from the mitochondria into the cytosol, along with those caspases. Telomerase, remember what this does? It's going to add DNA to the ends of it. What cells have telomerase? Germ cells, right? Those are the big ones. So I think a few other ones, but germ cells and a lot of stem cells. Not all the cells in our body. Over time, what happens? The telomeres get shorter and shorter and shorter. Keep in mind, if they get too short, what do we lose? Our important genes. And that usually there's a high correlation between decreased telomere length and aging. Or what happens if I have a tumor suppressor gene at the end of a chromosome and I lose it? Now I don't have that tumor suppressor. Now you can get unrestricted cell growth. You can get cancer growth, right? Something can happen there. Keep in mind the mechanism of telomerase. It's got an internal, template, right? That's RNA or DNA. RNA template. And it's going to extend what end of the DNA? The 3' end or the 5'? All right, well, telomerase is a polymerase. What does that mean by definition? What direction does it add shit? Always. 5 to 3. So, knowing that, if I've got a 3' and a 5', from here, 3' in, 5' in, if telomerase was going to add 5 to 3, which way, we'll just look at this photo, which way is telomerase going to work? It has to, if this is the end of the DNA, it's got to extend something this way, right? So which of these is going from basically 5, so this is here is 5 to 3, right? So it would have to be moving that way. Is this the way it wants to go? No. So it extends the free 3' end, always still in the 5 to 3' direction. This is what I mean by you guys actually know this, but you guys don't think you know it. And it's not that big of a deal now, but next semester, the reason it gets so much harder is they're going to be tricking you all the time, or trying to trick you a lot more, and there's so much information that you're going to get into an exam, and you're going to be like, shit, I don't remember this exact thing. But it doesn't matter that you know the exact thing, you need to understand the general process. You actually know a lot more than you guys think you do. And that's the key thing that you need to know, especially for next semester. But yeah, so it's this free 3' end, because this is the one that allows us to go from 5 to 3 going that way as it extends it down. It's going to extend out that strand, and once I extend that strand, what can I then add then? New Okazaki fragments, right? I add more, and then go back that way, and it goes on again and again and again. Once again, that's from the first exam. That's why blank says it's cumulative, because like stuff like that. But you don't need to go back over this whole lecture. I guess be pretty comfortable with that. Microarrays. Keep in mind, microarrays are great because they look at what are microarrays looking at in particular? Protein levels or what? Or gene levels or mRNA levels or what? They're looking at the mRNA, look at the transcript levels. Once again, high levels of mRNA does not mean I have high amounts of the protein. Because what do we have in between there? Those interfering RNAs, those microRNAs that can destroy that. So these are looking at mRNA levels of those genes, and even though we don't give us an exact read on the protein levels, it tells us enough. It'll tell us, well, it'll give us a fingerprint of the cell. So if I have two different people with different cancer types that were the same cancer type, but I want to see, even though it's the same cancer type, which genes were mutated or which ones do I have higher amounts of this transcript versus that one, I can use these DNA or these mRNA microarrays to look at the different fingerprints of those cells and all the different proteins that are involved. In this case, the transcripts that make those proteins. Same thing we were talking about before. Clustering programs to identify level of mRNA in a sample. So this is just an example. I honestly would ask him if he wants to. This is an example for whenever the cancer type was. These were the 4 main ones where even though, what is this? Okay, for breast cancer, basically they did a big study on everyone with breast cancer, not everyone, but people with breast cancer, and they found that they were divided into 4 groups, where one group of breast cancer patients had a lot of Beza-like mutations, or these were the transcripts that were screwed up, or higher or lower levels. A different one had HER2, Another ones had lobular, other ones had ductal. Different proteins or different transcripts that are responsible for these different things. And they were able to create these different subpopulations based on those microarrays. I don't know if he wants you to know this as an example for breast cancer of 4 main groups or not. I don't know. I think it's just a study, but sometimes Blank likes specific shit. So this would be something I think he could do. But once again, I'd ask him tomorrow if you see him. Because I'm pretty sure in the article, some of this was highlighted in the little picture, I think. That's why I don't know why I'm little. But once again, just ask him if you don't really know. Like we talked about mRNAs for profiling. Once again, remember, this is a discovery approach. It's not a hypothesis approach. This, you don't have to say like, oh, well, if this, then not this, or whatever. You just throw in the data and you see what comes up. And you see what sticks and you can grab and look at that. What's the other example of discovery approach we talked about earlier on in this course for this exam that you need to know? GWAS studies are basically like that, where you're not saying, oh, I know it has to be this gene out of 21,000 genes. No, you say, I have no idea what gene it is. We're going to look at all the different SNPs, all the different alleles running with those SNPs, and then see what comes up. He has additional issue of stuff in the textbooks. CDK stands for the cyclin-dependent kinases. They're players in a lot of those signaling pathways. AP1, we talked about this a little bit. It's the transcription factor for interleukin 2. AP1, though, is going to have different subunits. FOS and Jun. Know that. These are 2 subunits of that protein. Whereas I could screw up one of those, it could screw up the overall protein in general. Cadherins, they're important for cell adhesion, so like how a cell can be sticky and hold on to another cell. So we're talking like even something like metastasis, maybe cadherins get mutated that allows the cell to leave one area and then stick somewhere else, things like that. They can also act as, they can also basically have another, like how integrins can be bound to another receptor. Cadherins can do a similar thing where they can be bound to another cell, another receptor lead to a change in there. And the last thing is loss of heterozygosity. So, up to this point, I mentioned that if we're just assuming normal Mendelian genetics, where you've got big A, little a, and this is the mutated one, little a is mutated. I was saying that it's very rare that you're born this way, and then you end up with that. Because you have to have the 2 mutations occurring absolutely randomly. Very, very rare. But it can occur. Or you can be born like this, and you can end up with this one getting mutated. Or you could be really out of luck and be born like that, and you have the mutation from birth, or the disease of cancer type from birth. But another very common way is actually basically this, that you're born like this, and then you become basically that, where this is like nothing. You just lose that. Or you're born like this, and you're super unlucky, and you lose one of them, and then this one gets mutated. All of those are possible. What loss of heterozygosity is, it's this idea. It is that you don't get your allele mutated, you lose the whole, as in you lose the chromosome. It's just gone. And this occurs during mitosis. So during mitosis, what separates? Homolog, sister chromatids. Sister chromatids. What separates during meiosis? Both. Right? Both during meiosis. Remember, meiosis is on this exam. That's your first lecture on this block. So what basically happens with loss of heterozygosity is this. Let's say you have this. It's a recessive disease. So you're still good. You've got a big A and a little a. However, let's say as you're growing, developing in your cell, you've got that. So this good one is going to be the blue version. This is going to be the orange one. So one came from mom, one came from dad. I shouldn't use Florida because we're playing basketball, but it is what it is. So at this chromosome, you've got, let's say that's the maternal one, and you've got the paternal one. We're just looking at one chromosome. Let's say it's chromosome 1. What normally happens? These are going to go, they're going to get separated, and everything's going to be fine, right? They're going to go both ways because they're going to be sister chromatids that are going to be separated. What occurs is that instead of that happening, when you pull these away, actually I didn't draw that right at all. I'm sure I'm confusing everyone. Because once again, what lines up with mitosis? What lines up? Just the sisters themselves. So what I actually have is I've got chromosome 1, this one, and chromosome 1, that one, right? That's what's going to be occurring, and they both get pulled apart separately. And if we're happy, I get one blue and one orange in each. What happens here is a non-disjunction, where instead of them all getting pulled apart separately, I've got this shit going on, where both get pulled one way and neither gets pulled the other way. So if that actually occurred, I'd end up with this. Let's say, I said, which one's the bad one? I said the orange. Okay, so it doesn't really matter. So the orange one goes apart normally. This one gets screwed up. So I end up with orange. In this cell, I get both of these together. And then in this cell, I get none of the blue and one of the orange. So what alleles do I have here in this one? I've got big A, little a. This still isn't, it's kind of screwed up, but in terms of having that disease or this phenotype, it won't, because it still has the dominant one. This one just has the A. That's why I kind of draw it like that. There's a nothing there and then the little a. So a non-disjunction will lead to a loss of heterozygosity. What it means is you've lost the heterozygous trait. Because normally you have 2 alleles, now you're down to 1 allele. So if this cell continues to go on and divide, this can eventually lead to unrestricted cell growth because I lost that normal, whatever that is going to be over there. So non-disjunction will lead to loss of heterozygosity. And that's all this is talking about here. And so when we typically talk about where we can end up downstream with these mutations in this cancer cell type, it's not always, it doesn't have to be mutations. It could be something like this. This is still dysfunction, but it's not by definition a mutation in the DNA. I've just lost half the DNA in this cell. So that's another way it can occur. And this actually is a lot more common as opposed to getting 2 mutations or even 1 mutation. Getting born like this and then getting the other one lost is I think it's more common in many disease examples. And that's that. That's cancer genetics. I'll be here for a while. And then I can answer any other questions you guys have on this or other classes in general. And then I'll see you guys on Friday and then a month break. So just try to work hard for the next 2 weeks, and then you can do whatever the hell you want over winter break, or unless you have to study for the MCAT, then you can't do anything. But, all right, also, if you're planning to take the MCAT, like next semester, I can't stress this enough, you don't need to get done with your exam in immuno and then take it and study for it, but don't push off studying for it over break. If you're going to take it next semester, you should and need to start over the break at some point. If you don't, it's going to be very, very tough. Just for people that are doing it.